Hi everyone, welcome to Chapter 3, Amino Acids, Peptides and Proteins. In this lecture, we will be discussing Part 1 of this chapter, that is, amino acids. These are the learning goals for this chapter. To understand and familiarize yourselves with structures and names of various amino acids. To understand and familiarize with structures and properties of peptides. Ionization behavior of amino acids and peptides, and methods to characterize peptides and proteins. In addition to this, you would also familiarize yourselves with the way you purify proteins. Proteins are the most abundant biological macromolecules occurring in all cells and all parts of cells. Some of the functions of proteins are listed here. Catalysis is a crucial function of proteins. Enzymes are proteins that are involved in various metabolic processes. Transporting oxygen in the blood is performed by the protein hemoglobin. Transporting lactose across the cell membrane is performed by the transporter protein lactose permease. When it comes to structure, collagen is a protein that is involved in connective tissue. Keratin is involved in hair, nails, feathers, and horns. When it comes to motion in your muscle cells, myosin and actin are proteins that are involved in muscle tissue, and they are involved in motion. As I said, proteins are the major functional biomolecule and are used for nearly everything, including transport, catalysis, energy metabolism, structure, and so on. Proteins are also polymers of amino acids. To generate a particular protein, amino acids are covalently linked in a characteristic linear sequence. What is most remarkable is that cells can produce proteins with strikingly different properties and activities by joining these amino acids in many different combinations and sequences. From these building blocks, different organisms can make such widely diverse products such as enzymes, hormones, antibodies, transporters, and so on. Also, protein sizes range from as few as 30 amino acids that are linked covalently to thousands of amino acids that are linked covalently. What are amino acids? Amino acids are compounds that has both an amine functionality as well as an acid functionality. Hence the name amino acids. Traditionally, they are called amino acid residues when they are on a protein. Each amino acid has the same architecture, and there are 20 naturally occurring amino acids. All 20 of these amino acids are alpha amino acids, which means there is an alpha carbon. They have a carboxyl group, like I said, which is why they call the acid, and an amino group bonded to this alpha carbon. They differ from each other in their side chains. The group R is the side chain, which vary in structure, size, and electrical charge, and which influences the solubility of amino acids in water. In addition to these 20 amino acids, there are many less common ones. Some of these less common amino acids are modified when they are on the protein itself. But in this lecture, we are going to talk about these common 20 amino acids, their structure and properties. You must have realized by now, by looking at the structure, that So the alpha carbon has 4 different substituents attached to it, and hence amino acids are chiral, or they have chirality, right? A special D or L designation is used to describe the chirality of an amino acid. And because of the tetrahedral arrangement of the bonding orbitals around the alpha carbon, the 4 different groups can occupy 2 unique spatial arrangements, and thus amino acids have 2 possible stereoisomers, and you should have known by now, right? Since they are non-superimposable mirror images of each other, the 2 forms represent a class of stereoisomers called enantiomers. And you know this by now, that there is the R and the S isomers when it comes to enantiomers. And because they have a chiral center, they're also optically active, and hence they also rotate the plane of polarized light. Now, how do you determine the amino acid chirality? And that is by using a methodology called CON. Now, when you have an amino acid written like this, and H is pointing down, not pointing towards you, right? And you would use the abbreviation CON, where CO comes from the carboxylate, R is the R group, and N is the amine group, right? And if you go clockwise and it is CON, it is an L amino acid, whereas if you go anti-clockwise, CORN, or counterclockwise, it is a D amino acid, right? And most importantly, proteins only contain L amino acids. Several nomenclature systems are used for enantiomers. Like I said, R or S, that is based on Kohn-Ingold pre-log priority rules, right? Or small d or small l, based on dextro and levorotatory, or plus or minus, right? The D and plus means clockwise, L and minus means counterclockwise rotation. D and L still used on a lot of older drugs. The D and L are discouraged because they can be confused with the capital D and capital L, the system used with amino acids alone. So when it comes to amino acids, we will be using D or L amino acids, and will not be using the small d and small l. All right? Like I said, all amino acids are chiral except glycine. Glycine is an amino acid where the R group is H. So it doesn't have a chiral center. It is diastereomeric. Now, special nomenclature, like I said, has been developed to specify the absolute configuration of the 4 substituents of the asymmetric carbon, which is this one, right? The absolute configurations of simple sugars and amino acids are specified by the DL system. When I say DL, it's capital D and capital L system. And this is based on the absolute configuration of 3-carbon sugar glyceraldehyde, a convention proposed by Emil Fischer, and hence Fischer projections, right? For all chiral compounds, stereoisomers having a configuration that is related to L-glyceraldehyde are designated as L. So if a configuration is similar to L-glyceraldehyde, it will be designated as L. And if the configuration is similar to D-glyceraldehyde, it will be labeled as D. The functional groups in this case of L-alanine are matched with those of the sugars, right? L-glyceraldehyde, by aligning those that can be interconverted by simple one-step chemical reactions. The carboxyl group of L-alanine occupies the same position about the chiral carbon as does the aldehyde group. For example, the L-alanine carboxylate is at the place of the aldehyde. Now, one of the reasons, because the aldehyde is readily converted to a carboxylate group by oxidation. Historically, the similar L and D designations were used for levorotatory, where rotating plane polarized light to the left, and dextrorotatory, rotating plane polarized light to the right. However, not all L amino acids are levorotatory, and the convention shown may not work for amino acids, right? So by Fisher's convention, by relating these amino acids to the 3-carbon sugars, we will be using this D and L convention for all amino acids. Now let's see the naming conventions of these amino acids, right? All amino acid side chains except glycine have one or more carbon atoms. These are named sequentially using Greek letters. Branch groups get a prime or a numeric designation. Now for lysine, as you can see, it's a carboxylate and it's an amine, and this is the alpha carbon. All the subsequent carbons have been designated by the Greek letters. Now, since this is alpha, this is beta, gamma, delta, and epsilon, right? And this is the R group that we have seen before. Now, if it's a branched chain, you will have additional designation of 1 and 2. Now it's time that we get to know these amino acids. Now I do not have to tell you this, but it is important that you understand and remember the names and structures of these amino acids. It would be much easier if you remember the structure of the R groups so that you can compare and contrast. And that's why we are looking at R groups here. Since most of the amino acid structure is the same, the R group or side chain is what imparts unique chemical functionality to amino acids. The 5 major classes of amino acid R groups. 1. Nonpolar, aliphatic, hydrophobic. There are 7 of them. Aromatic, 3 of them. Polar, uncharged, 5 of them. Positively charged, 3. Negatively charged, 2. If you combine them, 2 plus 3, 5, 5 plus 5, 10, 10 plus 3, 13, plus 7, 20. So all 20 amino acids are accounted for, but they are divided into 1, 2, 3, 4, 5 groups. The easiest one to remember, the positively charged, negatively charged, and the aromatic ones. The polar, uncharged, and non-polar may not be that easy to remember, but there are ways to remember these structures and names, right? And I'll explain it to you as to how you can do that. Let's look at the first group, non-polar or aliphatic and hydrophobic. So the first one is glycine. The second one is alanine. The difference is that for glycine, it's a hydrogen. In alanine, it's a methyl group. Then comes proline. Proline is an exception here. If you were to look at it, proline has this cyclic ring, right? Now the cyclic ring extends into this amino group. So the NH2 here is a secondary amine as compared to all the other 19 amino acids, which are primary amines, right? That's the exception here. Fourth one is valine, right? It's an isopropyl group. Then leucine, another methyl added to the isopropyl, isoleucine. Now the difference between these two are isomers. Now the isomerism is because the methyl is attached here for isoleucine, whereas for leucine it is attached here. And then comes methionine. Methionine again is different from others because it has a sulfur. So these are the different amino acids, and here we will be introduced to the branched amino acids, right? Valine and isoleucine, right? Valine is branched, isoleucine is branched, as well as leucine. Leucine is gamma branched, whereas valine and isoleucine are beta branched. That's the difference. Now proline, like I said, has a secondary nitrogen, secondary amine group, and is sometimes known as the amino acid. Now, these amino acids, aliphatic, nonpolar, are found in the interiors of folded proteins. Why? Now that's a question that you need to ask yourself and answer it. Right? I will be able to help you out with this when we talk about this later on. Right? That's an interesting question, and you know the answer because we looked at this in chapter 2. Right? Think about it. The second group is aromatic. Aromatic groups impart strong absorbance in the UV range. And there are 3 aromatic amino acids. The first one is called phenylalanine. Now, you know what alanine is. Alanine is with a methyl group. Now, if you replace one of the hydrogen with a phenyl group, it is phenylalanine. It's easy to remember, right? Now, if you add a hydroxy group to phenylalanine, it becomes tyrosine. So these 2 structures are easier to remember as compared to others, right? Alanine, phenylalanine, and tyrosine, where phenylalanine and tyrosine are aromatic amino acids. Tryptophan. Tryptophan is an outlier because it has an indole group. The R group is an indole substituent, right? So tryptophan is a very it's an outlier, and it's a very important amino acid. We'll talk about this. Tyrosine is polar because in the sense that it has a polar functionality, right? Well, phenylalanine does not have a polar functionality and is completely hydrophobic. Tyrosine can at times poke its head into the polar regions using its hydroxy substituent because this OH can hydrogen bond with water molecules and other polar groups, right? Tryptophan is strongly fluorescent while tyrosine is weakly fluorescent. So tryptophan is the only amino acid in proteins that is fluorescent. Now let's look at the third group, polar uncharged amino acids. There are 5 of them here, right? They have slight preference for the exteriors of folded proteins. Polar groups are often the site of hydrogen bonding interactions. Cysteine is involved in disulfide bonding. Asparagine and glutamine are analogs of aspartate and glutamate. Now, we'll look at as to what aspartate and glutamates are, right? Now, the first amino acid that I want your attention is serine. Now, what is the difference? Now, serine in this case has a hydroxy substituent attached to this methylene group, right? Now, if there were no OH and it's just H, it is alanine, right? If you replace that H with an OH, it is called a serine. Threonine is when you have another carbon attached and a methyl, and in this beta carbon there is a hydroxy substituent attached. So this threonine is a branched chain amino acid. The branch is that it is a beta branched amino acid. It's very similar to valine. The only exception is that that methyl group in valine is replaced by a hydroxy, and hence the name threonine. Cysteine is another amino acid where one of the hydrogen in alanine is replaced by a thiol or SH group. So the difference between serine and cysteine is that serine has an OH and cysteine has an SH, right? And cysteines are involved in disulfide bonding. And what is disulfide bonding? And we'll talk about this later on. 2 other amino acids, asparagine and glutamine. Asparagine has an amide substituent in its beta carbon, whereas glutamine has an amide substituent in its gamma carbon, right? This is alpha, beta, and this is alpha, beta, gamma. So its amide is CONH2, and hence the name asparagine and glutamine. What about positively charged amino acids? often found in the exteriors of folded proteins. Again, think about it. Why? Three different positively charged amino acids. One is lysine, another one is arginine, and the third one is called histidine. What is the difference? Lysine has got a long alkyl chain. At the end of the alkyl chain is a primary amine that is positively charged, right? Arginine, on the other hand, has got this 3 alkyl chain, whereas lysine has 4 alkyl chain, 1, 2, 3, 4, and arginine has 1, 2, 3 attached, and to the third carbon, or the alpha, beta, gamma, delta carbon is a guanidinium group, and hence it's called as arginine. It's positively charged. Histidine, on the other hand, is where you have an imidazole substituent, imidazole functional group attached to the beta carbon. Now, histidine has a pKa that is close to biological pH. It's about 6. Now, we'll talk about as to what the pKa is and what are the consequences of the pKa of side chains, or the R groups of amino acids are, and stuff like that a little later. Now, let's look at negatively charged amino acids. There are 2 of them, aspartate and glutamate. Again, found on the exterior of folded proteins, sometimes on the interior as well. Both have carboxylates. So aspartate has a carboxylate attached to the beta carbon, glutamate on the gamma carbon, right? The pKa's are very low, but very close. This has a pKa of 3.9, and glutamate has a pKa of 4.1. And mind you, all of these are pKa's of the side chains. Now, aspartate and glutamate, the names are pretty similar to asparagine and glutamine, right? And hence, when we looked at the previous slide, asparagine and glutamine were called as the amide derivatives of aspartate and glutamate. And asparagine and glutamine are uncharged, whereas aspartate and glutamate are negatively charged. One more property of aromatic amino acids is that they have UV absorbance, right? It's a common way, UV absorbance is a common way to determine the concentrations of protein in solution. And you learn about this when you do labs, right? We typically measure the absorbance at 280 nanometers. That's where the maximal absorbance of tryptophan is. Right? Remember, tryptophan has this indole moiety. There's a tyrosine and phenylalanine. And if you look at the absorbance spectra, right, the absorbance of tryptophan is really high at 280 nanometers. Tyrosine, not so much, and phenylalanine is very less. Again, reiterating the fact that aromatic amino acids absorb light in the UV regions, and proteins typically have UV absorbance maxima around 280 nanometers. Tryptophan and tyrosines are strong chromophores. Remember this, right? Now, concentrations can be determined by UV visible spectrophotometry using the Beer-Lambert's law. That is, A is equal to epsilon LC, where A is the absorbance, E is the molar extinction coefficient, L is the path length of the cuvette, and C is the concentration of the sample. And in this case, you will be measuring the concentration of your protein using the UV absorbance by incorporating the molar extinction coefficient and a specific path length. The amino and carboxyl groups of amino acids, along with the ionizable R groups of some amino acids, function as weak acids and bases, right? So they have at least 2 ionizable groups. Now, what are they? As I said, amino terminus and carboxy terminus, right? When an amino acid lacking an ionizable R group is dissolved in water at neutral pH, it exists in solution as dipolar ion or Zwitter ion, and this is called as a Zwitter ionic form. right? This is the non-ionic form where you represent amino acids as NH2 and COOH. And as a zwitterionic form, you express it as NH3 plus and COO minus. Now, a zwitterion can function as an acid is when your NH2 is not protonated, right? And this is where the zwitterion acts as an acid. Zwitterion acting as a base is when NH3 is protonated and COOH is not protonated. In addition, some amino acids have a third ionizable group. Now, if the R group were to have another functionality that is ionizable, then you would have an additional complication for this amino acid. In addition to COOH and NH3 plus, R would also be ionizable. Right? Every ionizable group has a pKa. Now, remember what I said, right? You need to be familiarized with pH and pKa. pKa, again, reiterating what I said before in the previous chapter, pKa is the pH at which a specific functional group loses or gains a proton, right? Now, if you were to look at this structure here, carboxylic acid, right? Carboxylic acid in this specific amino acid loses a proton and becomes carboxylate. And when it does that, the net charge becomes 0. The pH at which this specific functional group loses its proton is the pKa of that functional group. Right? At a specific pH, COOH loses its proton. NH3 plus, however, does not lose its proton at the same pH as this carboxylic acid. It requires a higher pH for NH3 plus, and at higher pH, NH3 plus also loses a proton and becomes NH2. Now, when this kind of a proton exchange happens, the charge of the molecule changes, right? Now, if you consider, for example, at a pH of 1 or 2, the amino acid in solution will look like this with a net charge of +1, right? At a pH of 7, right, the carboxylic acid would have lost its proton and become COO minus, and so the net charge is 0 because the negative charge equalizes the positive charge. If you increase the pH further to about 11 or 12, the amino group loses its proton and becomes neutral. At the same time, the molecule's charge becomes negative because there's a negatively charged carboxylate. So that's how amino acids function as, or amino acids have acid-base properties, and the ionizable groups can help amino acids to alter the pKa or pH. This slide shows the properties and conventions associated with the common amino acids found in proteins, right? Now, there are certain things in this table, it's table 3.1, that I want you to remember, right? You can have this table with you all the times, right? If you want, you can print it out and just paste it right in front of you where you sit, because there are certain things that you need to remember. Right? These are basic things. This is where you start. Biochemistry 101, right? The names of amino acids, relate them to the structure, and relate them to their 3-letter abbreviation and their 1-letter abbreviation. The reason is because when you look at protein sequences, a protein sequence is a sequence of amino acids, right? And that sequence is unique for unique proteins. So the sequence is usually written in 1-letter amino acids. So 1-letter abbreviation for amino acids. And these 1-letter abbreviations could be the starting letter of that amino acid, or it may not be. For example, glycine is G, right? alanine is A, proline is P, valine is V, leucine is L, isoleucine is I, methionine is M. Whereas phenylalanine is not P but F because P is taken up by proline. Tyrosine is not T but Y. Tryptophan is W. So there is this difference, right? In addition to this, I also want you to remember the pKa for R groups. Now, one of the pKa's listed in this specific slide is for tyrosine, that is 10.07. Remember that number. 10.00 is fine. The other pKa values for the carboxylate as well as the amine are quite similar for almost all amino acids. For example, the pKa for carboxylic acid is close to 2, right? And for amine is close to 9.6 or 10. So that's something that I want you to remember. Pi for these amino acids can be referenced from this table, and so is the hydropathy index. So remember 3 things. One is the name, the 3-letter abbreviation, and the 1-letter abbreviation, and the pKa. Now, getting back to the second part of the table, right? We're looking at polar uncharged R groups and positively and negatively charged R groups here. Again, if you look at the 1-letter abbreviation, serine has S, threonine has T, right? Threonine holds T as a 1-letter abbreviation for itself and not tyrosine and tryptophan. Cysteine, C. Asparagine is not A but N. Glutamine is not G but Q. A is alanine and G is glycine, right? And when it comes to positively charged R groups, lysine is not L but K. Histidine is H. Arginine is not A but R. And when it comes to negatively charged amino acids, aspartate is not A but D. Glutamate is not G but E. Again, now here this table is very important because it lists the pKa of R groups of some of the important amino acids. Now, all those that have a pKa for an R group are the one that are ionizable, right? Cysteine has a pKa for its R group. That means the SH group can become S minus, right? It can lose a proton. And 8.18, at pH 8.18, the SH group in cysteine, the side chain of S-cysteine, loses its proton and becomes S minus. That's what it means. At a pKa or at a pH of 10.53, the side chain of lysine, which is an NH3 plus group, right? It's a primary amine, loses a proton and becomes NH2. At pH 6.00, histidine loses a proton. Histidine, remember, has an imidazole side chain. One of the nitrogen is protonated and it loses that proton and becomes neutral above pH 6. Similarly, arginine has a pKa of 12.48. Aspartate and glutamate, important ones. Remember these pKa numbers, 3.65 and 4.25. Now you need to remember all these pKa values for side chains, whereas for the carboxylate and the amino terminus, you don't have to remember much because they're pretty close, 2.0 and 9.6. You just have to remember those numbers. That's pretty much what you have to remember, right? So again, reiterating the fact, what you need to remember, the name and the abbreviations. The 3-letter abbreviation is easy. You need to remember the name and the 1-letter abbreviation and the pKa of the side chains. So let's sum up. Ionization of amino acids goes like this, right? At acidic pH, carboxyl group is protonated and the amino acid is in the cationic form, right? And the charge, depending on whether the side chain of the amino acid is neutral or not. If it is neutral, then the charge is +1. At neutral pH, the carboxyl group is deprotonated, but the amino group is protonated, and the net charge is 0 for a neutral side chain amino acid. At alkaline pH, the amino group is neutral because it loses a proton, and the amino acid is in the anionic form because the carboxylate is still deprotonated, and hence has a -1 charge if the side chain is neutral. So remember this.